Hello everyone, welcome to the third session of Metabolism of Amino Acids. So in this session we'll be discussing about deamination, oxidative deamination, non-oxidative deamination, decarboxylation and urea cycle. So by the end of the session you will be able to understand what is deamination, what is oxidative deamination, what is non-oxidative deamination, what is decarboxylation and the aspects of urea cycle. Deamination. The term deamination tells us or gives us the meaning like it is the process of removal of amino group from amino acids as ammonia. Deamination may be either oxidative deamination or non-oxidative deamination. Oxidative deamination is a process of liberation of free ammonia from the amino group of amino acid which is coupled with oxidation reactions. It takes place mostly in liver and kidney. The purpose of oxidative deamination is to provide ammonia for urea synthesis and keto acids for variety of reactions including energy generation. Here, we need to understand the role of glutamate dehydrogenase enzyme. In the process of transamination, the amino group of most amino acids are transferred to alpha-ketoglutarate to produce glutamate. Thus, glutamate serves as a collection center for amino groups in the biological system. Glutamate rapidly undergoes oxidative deamination. This oxidative deamination of glutamate is being catalyzed by the enzyme called as glutamate dehydrogenase. Either NAD+ or NADP+ as coenzymes are being used. The conversion of glutamate to alpha-ketoglutarate occurs through the formation of an intermediate compound alpha-aminoglutarate. So we'll be discussing about the regulation of glutamate dehydrogenase complex. So over here in this particular slide I have abbreviated glutamate dehydrogenase as GDH. So we'll be discussing about the regulation of the GDH activity in this particular slide. Glutamate dehydrogenase is a zinc containing mitochondrial enzyme. It is a complex enzyme with a molecular weight of 56,000 each. Glutamate dehydrogenase is controlled by allosteric regulation, GTP and ADP activates this glutamate dehydrogenase enzyme, and the steroid and thyroid hormones inhibit this glutamate dehydrogenase complex. After ingestion of protein-rich meal, the liver glutamate level is being elevated. Glutamate is converted to alpha-ketoglutarate with the liberation of ammonia over here in deamination reactions. Oxidative deamination takes place in the presence of amino acid oxidase enzyme. L-amino acid oxidase and D-amino acid oxidase are the flavoproteins processing the FMN and FAD respectively. They act on the corresponding amino acids L or D, levo or dextro, to produce alpha keto acids and ammonia. Oxygen over here is reduced to hydrogen peroxide. So in this reaction you can see over here that L-amino acid in the presence of amino acid oxidase enzyme is getting converted to keto acid and liberation of ammonia is being seen. Deamination reaction is being seen. This particular reaction is catalyzed by conversion of FMN to FMNH2, and FMNH2 is being oxidized to FMN again. During the formation or the oxidation reaction, there is reduction of oxygen to hydrogen peroxide in the presence of enzyme catalase. So this reaction releases out the water molecules too. Of oxidation of glutamate to glutamate dehydrogenase. So L-glutamate over here is initially converted to alpha aminoglutarate, which is further converted to alpha ketoglutarate in the presence of glutamate dehydrogenase enzyme. And there is liberation of ammonia, that is deamination reaction. Non-oxidative deamination is a process in which the ammonia is being liberated from the amino acids without undergoing oxidation. So examples for this non-oxidative deamination include the deamination of histidine in the presence of enzyme histidase. Histidase acts upon histidine to liberate ammonia by non-oxidative deamination. The amino acid desulfhydratase enzyme over here, desulfhydratase enzyme, the sulfur containing amino acids like cysteine and homocysteine undergo deamination coupled with desulfhydration to give keto acids. So this is another example of this non-oxidative deamination. Coming to the third example of this non-oxidative deamination, it is like the amino acid dehydratase enzyme. Amino acid dehydratase enzyme, the compounds such as serine, threonine, homoserine are the hydroxyl amino acids. They undergo non-oxidative deamination catalyzed by PLP-dependent dehydrases. So this is another example for non-oxidative deamination. So the next step of this amino acid metabolism includes decarboxylation. The tissues like liver and the microorganisms of intestinal tract contain enzymes called as decarboxylases, which require pyridoxal phosphate as coenzymes. They remove the carbon dioxide from carboxylic group and convert the amino acid to its corresponding amines. Example over here, histidine is converted to histamine in the presence of decarboxylase and pyridoxal phosphate. Over here you can see there is removal of carbon dioxide or decarboxylation reaction. After transamination reaction, the transfer of an amino group to the keto group, ultimately to form a glutamate ion, and then deamination to remove the amino group, then the decarboxylation reactions. The amino group which is being removed out over here, as well as the carboxylic group or carbon dioxide which is being released, are being utilized in the urea cycle. Coming to the urea cycle, urea is the end product of protein metabolism. The nitrogen of amino acid converted to ammonia, which is toxic to the nervous system and is fatal to the body. Hence, the nitrogen should be detoxified to urea. The urea is synthesized in liver, transported to kidney, and around 20 to 30 gram of urea is excreted through urine for a normal individual. Urea cycle was first metabolic cycle which was proposed and was given by Hans Krebs and Kurt Henslet in 1962. Hence we can relate this urea cycle to Krebs cycle in carbohydrate metabolism. Let's discuss about the various steps involved in urea cycle here. The ammonia which has been released from deamination reaction will be used over here initially. The carbamoyl phosphate synthase 1 or synthase 1 of mitochondria catalyzes the condensation of ammonia with carbon dioxide to form carbamoyl phosphate. This step consumes 2 ATPs and it is a irreversible rate limiting step. The next step involves the formation of citrulline. Citrulline is synthesized from carbamoyl phosphate and ornithine. Ornithine is regenerated and used in urea cycle, so its role is comparable to that of oxaloacetate in TCA cycle. In TCA cycle, if you remember, the oxaloacetate is being utilized in the TCA cycle and is being regenerated. Similarly, over here ornithine is utilized to form citrulline, and this ornithine is being regenerated in the urea cycle. The conversion of ammonia to carbamoyl phosphate by carbamoyl phosphate synthase 1 and the conversion of carbamoyl phosphate to citrulline with the help of ornithine. All this particular initial two steps will take place in the mitochondria. So this citrulline is being transferred into the cytosol, and in the cytosol takes place the synthesis of arginosuccinate. Arginosuccinase enzyme condenses citrulline with aspartate. So over here, citrulline and aspartate are being condensed by arginosuccinate synthase enzyme or arginosuccinase to form arginosuccinate. This step requires an ATP which is cleaved to AMP and pyrophosphate. The next step involves the cleavage of this arginosuccinate to form arginine. So arginosuccinate cleaves arginosuccinase cleaves arginosuccinate to give arginine and fumarate. Arginine is immediate precursor for urea, and the fumarate which is being released over here in this particular urea cycle will go into the TCA cycle. The fumarate liberated provides a connecting link with the TCA cycle, that is gluconeogenesis. Arginase is the enzyme that cleaves arginine to yield urea. And again, ornithine which is being yielded will be helping in the formation of citrulline by combining with carbamoyl phosphate. Or this ornithine so regenerated enters the mitochondria for its reuse in the urea cycle. Arginase is activated by carbon dioxide and manganese. The energetics of urea cycle include, like, urea cycle is an irreversible cycle and it consumes 4 ATPs in whole. 2 ATPs are utilized for the synthesis of carbamoyl phosphate. 2 ATPs are hydrolyzed to AMP and pyrophosphate to produce arginosuccinate. And over here we have the overall reaction of urea cycle, where ammonia and carbon dioxide, ammonia from deamination, carbon dioxide from the decarboxylation reaction, in the presence of aspartate and utilization of 3 ATPs gives us urea, fumarate, 2 ATPs, inorganic phosphate, an AMP and a pyrophosphate group. This is how the flow of nitrogen takes place in the amino acid metabolism. Initially, alpha amino acid is getting converted to keto acid or alpha keto acid in the step called as transamination. Later in transamination, the alpha ketoglutarate which is being formed is getting converted to alpha ketoglutamate. So this is D or deamination reaction where ammonia is being released out. There is decarboxylation reaction also which is been taking place, and the ammonia by deamination reaction and release of carbon dioxide by decarboxylation reaction combines or cleaves together and undergoes various steps over here in urea cycle to form urea. And this urea is being excreted through urine. So we'll have to remember that only way the nitrogen is getting excreted or the metabolized nitrogen is getting excreted out of the human body is through urea in urine. So we are towards the end of this session, and by the end of this session you are able to understand, I hope, what is deamination, what is oxidative deamination, what is non-oxidative deamination, the decarboxylation reactions, and the urea cycle. Thank you very much. Namaste.